So we have been talking about nanoscale devices, and we have looked at nanoscale materials. In many applications, we have come across microfluidic systems and microfluidic chips. But there is something that we need to know, that when we talk about fluid flow at nanoscale or microscale, there are issues, there's a different kind of equations or system approach that we need to adopt. So we'll talk about fluid flow at micro and nanoscale and look at a couple of examples that go with that. So now this fluid flow is a very mechanical engineering thing. So they study a lot of fluid flow, by the way, than us. But now when we deal with microfluidics or fluid flow, then we have to know what are some basic frameworks, basic equations, and how do they modify, and what transpires when we look at the fluid and suspended things in fluid, right? Suspending things being biomolecules. So that makes it interesting to see how the shear stress or shear forces will come into contact. And then when you look at solid, it takes much more force to deform it, but we look at fluid, it takes less force to deform it. So that force is what becomes interesting for us, and especially when we are moving the fluid around in narrow channels. At large scale, at macro scale, we use fluid to do many things. We may be moving air, we may be moving liquid to cool things, like for refrigeration. We may be moving fluid to transfer force from one side to another side, which is the thing in hydraulic systems. We may be using fluid to create thrust, like they do in engines. We may be doing moving fluid to transport materials. So we do all of those things at large scale, and there are a specific set of equations. Most importantly, they're called the set of differential equations called Navier-Stokes equations. So these are partial differential equations which gives us the motion of fluid, especially viscous fluid. Now, those equations, they have the fundamental premise that they conserve momentum and mass when we talk about the fluid, especially Newtonian fluid, right? Where the forces and the reaction of the forces, all the Newton laws of forces are conserved. And many times we have notion of pressure, temperature, and density also with that. But when we bring it down to microscale, we do similar things, but now the rules change. The rules change because now we have the forces that we ignore at macro scale, they become more significant at micro scale and below. The forces that are more prominent at micro scale, at macro scale, large scale, become insignificant at micro scale. So if I ask you, which is the largest force that acts on large things, on us, for example, what is the largest force that we cannot get out of?
Gravity.
Gravity, right? So
gravitational force.
Gravity, rotational force, exactly. So, but when you go below 5 micrometer, gravity becomes insignificant. At large scale, we are not facing charges. We almost ignore charges, but when we go below 5 micrometer, the charges become very significant. So we do many things, similar things at micro and smaller scale, but now the rules change. And the first thing that I would show is in nanofluidic systems or microfluidic systems, we don't know a lot of things even now, because the behavior is different. We know a bunch of things like charges, the effect of charges that would be there. For example, in this, if you see the above picture, that's a macro scale, large scale confinement. And we see charges are fairly distributed, especially in the middle. And the middle part is a large part of the overall fluid flow through that channel. And if you see the electric potential, that dies down in the middle. So more area is where the ionic concentration is uniform, like it would be in a bulk solution. But when we go to nanoscale, which is the nano channel shown below, what's happening here is the effect of the electric potential from the surface does not die down even in the middle, which means there would be going to be effect on the ionic concentration. So if the surface is positively charged, that means it's going to repel positive charges, and it's going to have much more of negative charge. So if you are pumping sodium chloride and you have a negative charge on the wall of that nanochannel, negative charge means it's going to attract more sodium channel inside the fluid flow. It's going to repel chloride, right? So that makes it, that's where the equations break down. Now, that also means that we can have an idea or a concept of forward biasing and reverse biasing now in a fluid flow, just like we have it in a diode. But here, this forward biasing or reverse biasing would mean more that we are applying a specific charge on the walls of nanochannel to get either fluid flow or stopping a fluid, right? So we can have kind of selectivity. So which means that when we go down, many of the macro assumptions, they break down. Like we take the fluid as a continuum at large scale, which means we take it to be one homogeneous entity. We also use the concept that the fluid sticks to the surface, so there is no slip. So there is resistance at the surface. It doesn't slip. We also use something called Reynolds number. So Reynolds number implies there is a relationship between the density of the fluid, the flow speed, the linear dimension, the dynamic viscosity. I'll show you what are all these parts that go into Reynolds number. So Reynolds number just gives us the inertial forces divided by viscous forces, which would tell us how easy would it be for a fluid to flow. When we go to low Reynolds number, that's where the microfluidics or nanofluidic flows come in. And the reason is the size of the channel becomes more than the molecular interactions, Brownian motion. Things are moving and they would bounce off of other things. So if there is an atom, it can bounce off other atoms and keep on changing direction. So we say overall flux of a Brownian motion is zero. So what if there are X number of atoms going to the right side overall, the same number is going to the left side. And then we apply voltage and we say more are going towards right or left. But here now, the size goes small, so the length of the passage of a Brownian motion atom or a we can say entity is smaller than the dimension of the container itself. And that gives us a new kind of way where at low Reynolds number, we have higher mobility. So the higher viscosity, which means it's much more dense. If you have, the example is if you heat up good, right, molasses, it becomes a very viscous solution. And if you compare it with water, water flows much easily, but molasses or the molten sugar would flow much at smaller speed. So that's the kind of thing that happens in microfluidic channels. The flow rate goes down because now you have a smaller volume. The volume is the same, but you have a smaller channel and things have to go through. But when that happens, it comes with an effect, what we call nozzle effect. So then the pressure goes up. So when all these things come together, we see that the fluids, they become laminar. So it becomes very hard to get a turbulent flow. Now, this is again a little bit something from mechanical side. The difference between turbulent and laminar flow is how things travel when there are fluids flowing or different fluids are flowing. So this example with colors is showing that if you start in a microchannel with 4 or 5 different kinds of fluids, they're going to maintain their movement, and they're not going to get mixed up because of low Reynolds number and what we call laminar flow. Very precise, very good laminar flow. But that's bad for us because in microfluidic channels, we want to mix things up, right? So the idea is we want to do things what we do in a test tube. So then there are more things that needs to be done and many different techniques have been done. So this is a real example of fluids which are flowing. And the upper side is the dark fluid, the lower one is fluorescent fluid. They keep on going in their own direction. They don't mix. The only mixing you see at the boundary is just because of diffusion. So this has been modeled, this has been shown, and many techniques have been applied to make fluids turbulent, so we can get mixing easily and hence achieve a reaction, whatever you want to do. So there's a little bit simulation work that people have done where they see that diffusion distance goes low. So which is x square is equal to 2 dt, kt over 6 pi efficiency and the area of the channel. So where they're showing that different particles, if passing through a microfluidic channel, will keep on following their track, keep on following their own area of fluid flow, except there'll be some diffusion that will occur at the interface. And they model different molecules and they show that the diffusion rate is very, very slow, micrometer per second. Whereas if you do that in a larger microscale channel, we see much more mixing or much more turbulent flow. Now, how does that transpire? It transpires into when we do devices where we are pushing fluids, we have to then apply more than just pressure-driven flow. Because pressure-driven flow would give you a parabolic kind of flow, again, laminar. But what we want is we want to mix things up. So what we do is then we apply electrokinetic flow. Electrokinetic, there are 3 types of electrokinetic flows: electrophoresis, electroosmosis, and dielectrophoresis. And we'll discuss them in a little bit. So what they do is then they help us mix things, or they give us control over the flow of things going through a channel. Here's an example of a nano device. So if you have a fluid flowing carrying charged particles, that can be a DNA, that can be any protein, a charged body. So the flow will be the combination of electrophoresis and electroosmosis. Now, in electrophoresis, the charged solid particles, they move under the external electric field. And of course, when they move the fluid with it as well. But in electroosmosis, the liquid with charge in it, that just the liquid moves under the electric field. And the charged solid stays stationary. So just fluid flow. So if you want to separate out the way we have done there in yellow and red, the electrophoresis is causing both solid and liquid to flow, to be separate out under the influence of electric field, whereas electroosmosis will only affect on the liquid particles, the ions in this example, under the influence of electric field. Now, the dimensions show that on the surface, for example, this is shown as oxide. The charge of oxide will influence the ions of that fluid to readjust. So there'll be, we call it electron double layer. What's written here as 2 parts stern layer and diffused mobile layer. So these are called EDL, electron double layer. And if the channel becomes too small, and if EDLs from both sides, they touch, that's where the fluid flow becomes difficult. Because now the external field is going to cause the gating effect. Right? So now they cannot freely flow. The lower picture is showing the pressure-driven flow and electro-osmotic flow. So we see the difference. Pressure-driven flow has a parabolic shape, but electro-osmotic flow would flow everything together, right? Because that's where the charges are making the external field is causing all the liquid particles to flow in that direction. But now when we do the functionalization of these surfaces, the problem becomes even more difficult. So now if you put a molecular recognition element like DNA molecule or a protein, this becomes even more difficult. So molecular SAM brings its own charges, brings its own issues inside a microchannel. Now, what do we do with that? So this has been used to our advantage by using the molecular recognition of those molecules. We have been used to achieve what we call active filtering. Right? So we talked about that before, where we can now make the channel distinctly see what molecule or what species can pass through much more than the other entities. So we can now imagine, so this becomes, if you want to analyze this system, it becomes a little bit difficult because now there are many more charges involved. But in this case, then we take, we learn from biology, we learn from living systems. In living systems, we have what we call nanopores. They're called ion channels also. So what ion channels do is, here's an example on the right side. It's an alpha hemolysine ion channel. So ion channels exist in all cells, right? Cell walls, nuclear walls, they are there. And their job is to pump specific ions across the boundary, across the nuclear wall or across the cellular wall. What does that mean? That means if we have sodium channel, that protein has only one job. It's going to be pumped sodium ions across the boundary. So in this case now, instead of looking at each individual pore and what's happening inside the pore, we are looking at overall job or overall physics of the channel, a composite kind of effect of the whatever is inside the channel. And lots of work has been done. I will get into that. But let me just introduce ion channels and nanopores. So ion channels, what we now call biological nanopores also, but biologists still call them ion channels. So they exist. There were reports about, I think 20 years ago, where they isolated ion channels, they suspended into a lipid bilayer, just like the cell wall. And they were able to use these ion channels to detect single DNA molecule passage. And this is one of the early reports from Harvard. What they did was, now when a DNA molecule passes through, so they had the electrode set up on both sides, and they had ionic current flowing because of the potential. So you could measure current, which is shown on the right-hand side, with time. So trace of current versus time. And then they introduced DNA molecules. So DNA is negatively charged. They introduce it in the positive terminal side. Sorry, the negative terminal side, which means now the positive charge on the other side of the pore. So ions are flowing through the channels anyways because of electroosmotic flow. But now, as soon as a DNA molecule comes close to the mouth of the ion channel, it gets swept to the other side. And that passing of DNA molecule was observed as the blockage of the ion channel, which showed up as reduction in the current that was being measured. So spikes, downward spikes, which is shown on the right-hand side picture. And those pictures, those spikes, when they zoom them out, you could see specific behavior between them. Behavior means the blockage, how much the blockage was, how deep was the blockage, was seen to be characteristic of different bases. And there lots of work has been done to get there, but ultimately this is what has been achieved, that possibly we can read a single base as it goes through. I said possibly because there is still debate whether we are looking at single base or we are looking the effect of multiple bases that are going through, just because the bases are so close to each other. G and C and A and T in a long chain, they're just how much, what's the distance between base to base?
Sir,
yeah.
सर एक क्वेश्चन है सर, जो डीएनए पास करते हैं सर नैनोपोर से सर, वो करंट जो आयनाइजेशन करंट सर उस पर डिपेंड कर रहा है सर. अगर वो करंट ज्यादा था सर तो वो डीएनए की सर शेप किस तरह हो, मीन फिर भी
you have to
डिफेक्ट होगा सर.
hold on. So current is not because of DNA. Current is already flowing. You have applied electrodes and ions are flowing. And then you introduce DNA and DNA starts flowing as well. And as soon as the DNA starts going through the ion channel, the current is blocked. And that current blocks shows up as reduction in current. It is not current due to DNA. It is current because the channel is getting blocked from DNA passage, right? So, but there is more coming up where we could be able to see the charge that's carried by DNA as well. So just stay with me. Now, so these nanopores, the lower picture is showing a synthetic nanopore made in silicon. So they've been used now to do a lot of characterization work where we have, so we can see DNA interaction, we can see DNA protein interaction. We have been able to see secondary structures in the DNA molecules like hairpin loop. We can measure the binding of nanoparticle with the DNA. We did some work where we could hold a protein or a nanobead right on top of a nanopore and could measure charge, like fundamental 1 electron charge on 1 nanobead. So there's lots of work that has been done. The beauty is that the interface is at nanoscale, but you are measuring a current in the outside system. So you can say you can measure current at macroscale. So the solution that we use before we introduce DNA is always an ionic solution. You can use sodium chloride, for example, but we don't use sodium chloride, we use KCl, potassium chloride, because it is the lipid bilayer molecules are more stable in that, and the current is also more, there's less noise in there. So they've done more interesting things as well, but this is just breaking down that as the DNA passes through that ion channel, we get the dips, and those dips are different. So some dip or pulse would be smaller in weight, bigger in weight. Some dips would be deeper. Some would be less in depth. Some would be longer. So there are a whole bunch of activities we see in the data that helps us cluster different molecules based on their behavior as they're passing. So this picture is just showing the behavior, that how we can distinguish between the 2 types or 3 types or more types of molecule as they're going through. So width versus depth can help us distinguish or cluster, mathematically cluster. So we can apply some of those statistical analysis on the data and separate out different subpopulations, which we then can call, like they have done here, that the one is A or one is T or one is C, or all those different bases have been shown. So, all right. So, and then more work has been done where they, instead of just using an ion channel, they put a sequence in it. they bound a sequence. For example, this seen blue DNA molecule. So now that showed that if the solution had complementary DNA molecule, the pulses were different, because now as that target sequence passes through, it first binds and then it has to gets released and passes through. So which shows the difference in data between complementary and single base mismatch. So that's like the most fundamental limit of what we can measure, right? So if you see the left one is showing perfect complementary target molecule in solution binding to the probe molecule, which is bound to the nanopore, and you see the data shows different behavior between those two. So there are many more things that have been, and the lower example is showing where they used an adapter and used another biomolecule which could bind to the things that were passing through to show the behavior that was different. So this was taking pores from living systems, but as far as 89 people were making nanopores. They're making to just study different things, but not for bioapplication. But that started in about 2004. And I was in the middle of this thing when this started. So the idea was that you take a silicon wafer, you etch through, you get to a thin membrane, and then you make a very small hole into that using, there are many ways how it's been done. We have done with transmission electron microscope. So we focus a thin film, we focus that very tightly focused electron beam into a thin film, and that causes the oxide or silicon to melt and create a small nanopore. So this gentleman Chang, Dr. Chang, he was postdoc in my lab later on. So we did lots of work on this. So the idea is now, so you have, so this lower picture, you just turn it sideways and you think about, we put ionic solution on both sides and start measuring the current. So we get a trace of current again with respect to time, but then we introduce DNA molecule, and as soon as that goes through the pore, we see a dip in current, and that dip in current is what we measure as signal from the DNA. Again, similar signal, the upper figure is showing that there. So we took that nanopore idea to apply for DNA detection. So there's interesting phenomena that has been seen. So ideally, what we would think that when DNA goes through, there should be a blockage of current, but we saw something else as well. We saw that in some cases, we were seeing a pulse, upward pulse, which meant that the current was increasing when the DNA was passing through. So that meant that it was not just blocking, it was doing something else. It was modulating the current, it was increasing the current. And that, it was published a while ago that what we were seeing was, we were seeing the DNA molecule has charge around it. So that charge, when the molecule was going through, the charge was contributing to addition of the current. So that is kind of a opposite to what generally is believed. And then there's so many things have been done. There's people have seen the folded detection of folded DNA. People have done the detection of comparison between a double strand and a single strand. So a whole bunch of things have been done since then. But now, if you look at the conductivity of one nanopore, you can break it down into three pieces. So this is what this equation is showing. We have broken down different parts of that current that will be flowing. So one is just the ionic current, right? Resistive part to it. Another is the movement of charge, ions, so ionic current. And then there is the contribution of DNA molecule, which is carrying charge with it. So all these three entities come together to give you an overall current of the pulse. And that's what is shown here as I DNA, I counter, and minus I block. So the blockage current. So now what we are doing here is we are looking at fundamental charges, very, very fundamental charges using a nano device, right? So this, there's much more to it. We can go into depth if we would like to, but just today I'm just touching upon this. When you talk about the resistive nature of the pore, that can be modeled just like a resistance for what we call Ohm's law, V equals IR, because for a pore, the ions and the voltage, you increase the voltage, more ions will flow through, which means the current will go up. And that has been also shown that now the fundamental relation of resistance with mobility, with conductivity, rho L over A, or resistivity. So we use 1 over gamma, or 1 over conductivity is equal to resistivity. So here then the resistivity can be now taken in terms of fundamental charge transfer from solid state devices, which is mu Qn, right? So what the idea here is that if you think hard and you can understand all the features going on, we can break down into simple physical terms and understand what's happening. Another thing that we did was using a nanopore and attaching a single strand DNA to achieve selectivity, just like they did for a bio pore, for an ion channel. So here what we saw then, the perfect complementary sequence, when that was going through, number one, there were many more of the molecules would be going through, and number two, the pulse were much deeper, which showed that the binding was causing bigger blockage of the nanopore. So this was done with synthetic nanopore. And then we looked at the physics of that, and we found out that this was just fixed first law of diffusion. You know, diffusion is governed, we have model of diffusion. And in this case now, the pulses were changing because the tau here, the time, the resident time, which is n over t, that was more, and more means the flux would be smaller, or smaller would mean flux would be more, right? So it's a reciprocal interaction. So there's, again, you have to look at the binding mechanisms, you have to look at the friction, you have to look at the resistance, the mechanical parts. So all those equations come together to give you a control or a model of what's going on. So it's again looking at overall ion channel, we look at the potential across the channel, and we look at what is the contribution to the potential by the binding events. So again, a little bit more involved. All right, so now we have used that also to create active filters, right? So we're talking about filters. Filters generally at micro world, in macro world, filters are used to separate out bigger things from smaller things. But when we talk about active filtration, we want to separate out specific molecules from other molecules, irrespective of their size. So what was achieved is shown in cartoon, that there's a whole mixture, red, green, blue, but what we want is we want to enrich the green things on the other side. So again, similar physics goes into that, where the binding causes the overall flow across the membrane or across the channel much more than the other thing. So this is now mimicking ion channels. So as we were talking about sodium channels or potassium channels, who would pump one kind of entity more than the other. But now we are implementing that using synthetic materials. So the data we showed with data that, yeah, the binding would result into more flux for example, it's showing hemoglobin versus BSA. So you could see more flux with that. All right, any questions?
Sir,
Yeah, go ahead.
सर जो हमारे साथ बैक्टीरियल डीएनए होते हैं सर, क्या हम भी इसी नैनोपोर से पास कर सकते हैं सर?
So yeah, so it's DNA. At the end of the day, it's DNA, right? So if you can, now the question would be selective detection to find that if that sequence is going through, which we know belongs to a viral load, yeah, we can detect the viral load in a solution using functionalized nanopores.
सर दूसरा क्वेश्चन ये है सर, इस प्रोसेस के लिए सर हमारे पास आयनिक करंट भी पास हो रहे थे, उसके लिए बकायदा एक सिस्टम चाहिए सर उसको मेजरमेंट के लिए। तो बनिस्बत है सर जो एल्यूमिनियम सीक्वेंसिंग है वो बेटर है सर, यही नैनोपोर
So
सीक्वेंसिंग बेटर है?
sequences is something which where you, okay, now sequencing requires certain steps, right? So where you have to put enzymes, you have to run the gel, and there's limitation of how much you can run in how much time. But here what we are talking, you take the sample, you run it through in 1 hour, you have the answer. Right? So sequencing is gold standard, but what we are trying to do is make things fast and make things cheaper.
मेन सर नैनोपोर सर कॉस्टली प्रोसेस सर बनिस्बत है सर।
So once you set thing up, what we are doing is there's unlimited opportunities with that. Right? So you know the sequence, you functionalize with the sequence, and you get the answer.
सर इसके डिवाइसेस मार्केट में मौजूद नहीं सर सिवाय ऑक्सफोर्ड नैनोपोर के सर?
So Oxford Nanopore is also not available in market.
या लेबोरेटरी तक है सर?
Right? So I know the team, I've read their proposals also. So that's also, yeah, there's some of the things are still debated. But there's a nanopore device which was proposed for Mars mission also, because then, so that had a scope very different, where you wanted to look at chemical nature. So use it more as a spectroscopic tool than looking at specific DNA sequence. So it has many other applications apart from what we are talking here, because the beauty is that you have a nano interface which gives you a micro measurement.
सर लोगों ने इसके बहुत प्रपोज सॉल्यूशन दिए बट कोई भी इंप्लीमेंट नहीं हुआ सर, इसकी वजह क्या है सर?
So that's where you have to know what are the missing components or what are the challenges. So I know many challenges which still exist. So the fundamental question is the bases are too close. So your membrane has to be really thin. So graphene can do that, but then the signal that you're measuring has to be at a very high frequency, right? So you can measure one base passage at a time. So electronics, so there are many questions which still need to be resolved before you can come up with a technology solution.
So this solution is only for the DNA molecules?
No, it's been done for many things.
Protein,
Many entities have been done with this. Biomarkers, nucleic acids, proteins, single electron charge using nanobeads. So measuring one electron charge difference between entities. All right, so micropores. I talked about this in a webinar I gave a few days ago, so I'll just brush through quickly. So micropores now are bigger, right? So if nanopore is 2, 3, 5 nanometer, micropore is 1,000 times bigger than that. And why do we want to use this? To look at cells at cellular level. So don't mix nanopores and micropores. Why together? Because we are talking about fluidics, flow at micro and nano channels. So many things have been done. Abil can share a video where I've shown, as they're going through, you see the similar idea of current blocking. I owe you that. I have to sit with you and show you how I made that. But anyway, so
सर आप बता दें सर, फ्यूचर के लिए हमारे लिए बेस्ट होगा सर?
it's straightforward. We'll talk about that. We are online. Let's stick to the material and then we can talk about stuff. All right. So when we get into macro channel or microarrays, similar problems exist, but now we do more of pressure driven flow. So we don't come into the problems of electroosmotic flow, but we still have to think about how much charge does a cell carry and how that would impact your measurement. So Micropores, we create them and we sandwich them into a measurement system. So this is the fabrication process going from A to F. And then you package it into kind of a block where now you have microchannels attached from both sides, and you can optimize the flow depending on what cells you're measuring and how quickly you want to process all of them. So there's a lot of work has been done where we have discriminated cancer cells from normal cells, where we have discriminated one type of cancer cell from the other type of cancer cells, where we have been able to clearly distinguish between metastatic cells and non-metastatic cells from the same tissue, where we have been able to classify there's in one tumor, there is heterogeneity. There are many different types of cancer cells. So we have been able to see 5-6 subclasses of cancer cells based on data from micropores. So this is a much bigger field, but it is at microscale. All right. So we'll get into a little bit more on